shall we? Of course, you guys probably like something to look at. About right. So we last left off, we were talking about some of the enzymes involved, right? So we talked about primase, that was number 6 on our list. We have a few more to go. But before we do that, because we've been talking a little bit about continuous and discontinuous strands, right? So what we want to do is take a look at DNA pol 3 and take a look at a particular component of DNA pol 3 that's associated with it. So this is the easy part, right? So this is your leading strand. This is your continuous strand. Basically, all you need is one priming event, and then your pol 3 will be able to bind to that area and then and kind of move along. Well, here's the question, right? How does DNA pol 3 bind to that 3' hydroxyl group, right? So what's the mechanism of that? Well, as it turns out, that in the pol 3 complex, we noticed that there was subunits called the beta subunits that actually formed what's referred to as a sliding clamp. And so here you can kind of see the sliding clamp's job is essentially grab onto or clamp onto the DNA, and then basically it slides along the DNA. So literally it's kind of like taking like a pen like this, the clamp will just basically clamp onto that particular area and just slide right down the DNA. Now, of course, that's one part of it. The other part of it is the clamp is also attached to pol 3. So the clamp itself is kind of like the motor that kind of guides the pol 3 along the DNA strand, okay? And so once you do that, then you're off to the races. And it's a fairly smooth process if you're on the leading strand. Of course, we now know that the lagging strand has issues, right? So it's discontinuous in nature. So what are some of the things that are going on the lagging strand side of things? Well, obviously you have pol 3 because that's the workhorse, right? That's the one that's laying down the majority of your nucleic acid. Your primers are being laid down by primase, right? That was the last enzyme we got introduced to last time. And then, of course, it's this primase that is kind of creating these Okazaki fragments, these little chunks, if you will, which are then removed and replaced by DNA pol 1, right? So pol 1's job is to go in there, remove those primers, and to replace it with DNA. So your DNA is not a patchwork of DNA and RNA, right? Because the RNA components, those primers, are removed from your DNA molecule and replaced with DNA. So your DNA molecule is all DNA. Right. Now then, here's the thing though, the important thing to understand about polymerases is all polymerases do is basically do dehydration synthesis. That's all they do. So they basically take a look at your template and then they pair up the corresponding base with that template and they go through a dehydration synthesis. So what happens when you run onto that last base in sequence? Like for instance, when you're a lagging strand, you've got this chunk that's already made. You have your primer right there. And here's your DNA polymerase, which is coming along here. It's laying down these pieces, but now it kind of runs into that little chunk that's in front of it. Well, why doesn't it just go ahead and seal it up? Because that's a different chemical reaction. Remember, the polymerases can only add stuff to it. Sealing the nick in the backbone is a different reaction. It's not a dehydration reaction. Okay, so all the polymerase can do is continue to add. So if it did that, what would happen is it would basically keep adding and adding and adding and adding and adding, and you kind of get this big old dangly piece over there. So it can't actually connect or abut up to that other fragment. There's a different enzyme whose job is that, and that's DNA ligase. So DNA ligase, basically its job is to seal the nicks in the backbone. That's all it does. All right, so it just goes in there, finds those nicks, seals it up, which is the reason why it's not a big deal for topoisomerase to go ahead and nick the backbone to really torsional strength because you have DNA ligase who can come in and seal those nicks up. Okay. So if you're keeping track, DNA ligase is the 7th member of the cast that we've met. And then, of course, you have DNA gyrase, which, like topoisomerase, is kind of a smoother outer, for lack of a better term, right? But it also is important in termination because DNA gyrase will actually unlink the two copies, kind of separate them. So it's kind of a separator in addition to that. So DNA gyrase has a couple of functions here. So let's take a look at this. This is kind of like a nice view of the lagging strand. It's nice and clean. So here you can see you have your initial primer. Your DNA polymerase 3 has largely filled in that gap. So here's your DNA pol 3. It's already fallen off, right? Because it's just run into the next fragment. It has nowhere else to go. And so it's already fallen off. And now what it's going to do is it's going to come back here and find its new primer. The primase is already laying down, right? So as soon as helicase is unwinding the fork, then primase is looking to lay down a new primer. So the DNA pol 3 is going to find that primer, and it's going to basically fill that gap in until it hits that next fragment, and it stops and it falls off. So notice that each of these fragments have a little gap in between them. Well, the next task you need to do up here to process this is to remove those primers and to replace them with DNA. And that's the activity of pol 1. So you can see pol 1 is actively replacing this primer and going to replace it with DNA. So it's all DNA at this particular point. But remember, pol 1 is also a polymerase. So this just lays down stuff. It doesn't seal the nick. So that's what ligase then comes in. So DNA pol 1 will basically replace the primer with DNA nucleotides, and then it'll leave the nick there. And then what happens is ligase comes in and sees all these little nicks and just seals them up. So you can see ligase is coming in here, just seal up that nick. And once ligase is done doing its job, then you've got a nice molecule of DNA. So that's kind of lagging strand in general. The replosome, remember we talked about the replosome. This is basically the big complex of all these replication components and all together, essentially. So you have a couple of different pieces of the replosome, this kind of gigantic complex. The first one is what's called the primosome. This is kind of like focused on initiation. Remember we said we had a three-step mechanism, right? Initiation, elongation, termination. Well, the primosome is associated with initiation. So you have primase, right, setting down the primer. You've got helicase, the unwindase, it's unwinding and exposing the template in the first place. You have accessory proteins. What are those? Single-stranded binding proteins and things like topoisomerase. So those are your accessory proteins, right, which basically are either stabilizing the single-stranded template in the case of single-stranded binding proteins, or smoothing it out, right, your smoothase, if you will, right? I love enzymology, right? You just create a word and put A's on the end of it. So these are all events of initiation. Of course, then we get to elongation, and that's where you kick it over to pol 3, and then you're basically doing leading and lagging strand at that particular place, at that particular point. Okay, so let's take a look at this. This is kind of like the scary version of it, but it has all the players in there. There's actually a couple of new people that we've been introduced to, and now we have to introduce you to the coordinator. So basically, when we're taking a look at the cast of characters, it's similar to basically taking a look at an orchestra. And what you've done is you've basically been introduced to all of the musicians in the orchestra. You've met the first chair violin, second chair violin, you met the oboist, you met the bassoonist, you met, the cello player, the violas, you met the individual musicians. But the person we haven't met yet is the conductor. The one who pulls all the musicians together and basically makes it all fit. That individual is the clamp loader, the squid in the middle, okay? It's for you oceanography people. So we have all of our pieces here. So let me just kind of highlight where we are, then we'll talk about the mechanism as we move forward. So your gyrase / topoisomerase, right? These are the smoothases, are moving out ahead of helicase, smoothing out that torsional strain in the DNA molecule. Helicase, of course, you can see is here. Primase is laying down its primer. You've got DNA pol 1 replacing the primer and ligase sealing the nicks. You also have the clamp that is attached to pol 3. That's this big guy right here, the big kind of peanut looking thing. And so that basically is moving in this direction. But now let's take a look at the clamp loader itself. The clamp loader, the ultimate conductor, is basically in contact with it all. So you can see that the clamp loader is tethered to the helicase. Helicase isn't on its own. It's being guided by the clamp loader. Same thing with primase. When primase is laying down the primer, it's attached to the clamp loader. So the clamp loader is actually actively guiding the primase activities. It's also attached to each of the polymerases, both on the lagging strand and on the leading strand. So the clamp loader is basically in contact with all of the major pieces of the primosome, even though we don't necessarily think of the clamp loader is like, oh, the clamp loader is it, right? Okay.
We need to kind of like the inside of cells should be locked this image for burning.
Yeah, there's several coming. There's several of these coming because this is your base image to understand the mechanism of how the coordination of leading and lagging strand is pulled up. Then, of course, you have your open beta clamp here, right? And of course, the squiddly part, right, the little tentacles, is what's called your clamp loader domain. So this is our beginning, right? So here we can see we've got a DNA pol 3 who is actively filling in this gap, right? It's removing single-stranded binding protein and replacing it with nucleotides. It's going to do that until it hits this next fragment ahead of it. You can see it's basically bound. And so what we want to do is we want to take a look at this. There's 4 steps to this mechanism. And so we're going to see how this basically works out. So here's the image we just saw, smaller version of it, but this is it. So your pol 3 is busy laying down your nucleic acid. It's being guided by the clamp, where the sliding clamp is actually sliding and kind of pushing the DNA polymerase along. But notice your DNA polymerase is also being guided by the clamp loader that has a little squiddly tentacle attached to the DNA pol 3. At this particular point, you notice the helicase has opened up the replication fork, exposing more template. And so primase has come in and is now laying down primer upstream. Of course, your single-stranded binding proteins have noticed the single-stranded DNA template. So they've basically bound to that in order to stabilize it, make sure it doesn't zipper itself back up. By the way, the leading strand, we don't even need to talk about that one because basically you're just plowing right on through, right? So there's really not a lot going on up there. But notice you're guided by a couple of things, by the clamp and also by the clamp loader, okay, which is guiding that process all the way through. So now let's take a look at the next step. So basically, once the DNA pol 3 finishes up, so here you can see it's finishing up these last couple of nucleotides. Notice at this particular point, the primase has finished, and so it's dropping off. So now you have an active functional primer ready to roll. Your open template is protected with single-stranded binding proteins. And not only that, but your helicase continues to plow into the replication fork and open up new single-stranded template. Now, at this particular point, what's going to happen is that polymerase is going to finish. And when it finishes, it's going to drop off. But notice what happens when it drops off. So when it drops off, this is the segment that it just finished right here. So when it drops off, notice it disassociates from the clamp. So your clamp releases. But it does not dissociate from the clamp loader. So the DNA pol 3 is hung on to by the clamp loader. Okay, so what's your next step? Well, basically you have ready to roll primer. You've got your DNA pol 3 here, and you've got your clamp. So now what you have to do is you have to assemble your clamp, attach your DNA pol 3, and you're ready to roll. But you have to assemble it where? On your new primer. Okay. So now what happens with the clamp loader is basically it will do just that. So you can see the clamp loader will basically grab onto a clamp. And it assembles it to the new primer with pol 3. So you can see basically it brings in the clamp with the clamp loader. So it kind of uses this little squiddly little tentacle thing to grab onto the clamp loader, pulls it down, attaches it to the new primer, and then takes its big long tentacle with the polymerase and attaches that to the clamp. So you basically have the 2 different parts of the clamp loader attaching the clamp to the DNA polymerase 3 to the clamp. So it's assembling it at this new primer And then once you do that, then you've got what you need. You've got a new complex on your new primer, and away you go. But notice what's happening. Helicase continues to move forward and open up new template, which means primase comes in and is going to get ready to lay down a new primer. And so it goes over and over and over again, as long as helicase is opening up new template until you reach the end. Okay. So that's basically the mechanism. That's the reason why I said, yeah, you want to know that image, because there's a stepwise mechanism of how this actually happens. So for you table people, this is pretty much your list of the major players, right? So if you can run down this list and explain what all these guys are doing, then you're in pretty good shape for this chapter, because explaining what these guys are doing requires you to explain a lot about how lagging strand works. And if you can explain lagging strand, then leading strand is an easy one. Okay, so that's all in prokaryotes, right? But what about eukaryotes? So basically we took the replication mechanism in prokaryotes and we realized that it's mostly the same. Mechanistically, for instance, the enzymes are very similar in nature. But we have a couple of functional differences, right? So for instance, we have a lot more DNA to deal with, and we've got linear, which means we have ends, right? Bacteria didn't have ends, it was a circle. So you just start it and you just run into each other and you stop, right? But now we've got ends, which is a problem. And we'll talk about that here in just a little bit. But for the most part, replication had a lot of carryover from prokaryotes to eukaryotes when we asked that final question. So a couple of the differences that we see is, even though we have origins of replication, in eukaryotes, we have multiple origins of replication. So, and not only that, but what we have is not so much replication forks, but when we take a look at a eukaryote, we'll actually see replication bubbles. Lots of them, all along the chromosome. So here's your multiple origins forming replication bubbles. By the way, when we draw the replication fork, all it is half a bubble. So in reality, a replication fork, if we were to draw it all the way out, basically is just 1/2 of a bubble. So what we do is we just basically look at this side. Okay. But it's the same basic idea. The other thing also is that in eukaryotes, our origin is not sequence specific, and in prokaryotes it is, right? So you have a sequence that says start here. But in eukaryotes, it doesn't seem like there's a sequence signal to start your origin. You just kind of start it in different places, and then it can be moved around, or you can slide around a little bit. So that's another big one. Oftentimes in initiation with eukaryotes, we also have a lot more going on. So we have a lot more replication factors, especially in initiation. A lot more things working with helicases and primases and things like that. Like, for instance, a good example of one of the differences in our primase is, remember, in bacterial primase, it's just an RNA polymerase, right? But in eukaryotes, we have both an RNA polymerase and we have a DNA polymerase version of a primase, which is unusual, but we do have one. There's also some structural differences in our polymerase itself, right? Our pol3, our workhorse enzyme. So instead of being constructed like bacteria, we have some major subunits in our pol3, epsilon and delta, which play a prominent role in the polymerase function. Now, we're not going to get deep into the woods on the polymerase. You know, we would if we were like in a molecular genetics class and we were getting into, like, had an entire chapter on replication. We were getting into the mechanism and the entomology of replication, and we'd ferret out all the subunits of eukaryotic replication, but that's not necessary for this one. But in eukaryotes, we've got these linear chromosomes, right? So that's a problem, because that basically means you've got ends. And the fact that you've got ends is a little bit challenging. It means you have to basically come up with a strategy for protecting those ends. And our strategy is to create what's called telomeres. So telomeres basically are the ends of chromosomes. That's what these are. That are a series of repeats. So it's just like repeated DNA, a repeated sequence over and over and over again. They act kind of like adapters. And what they do is they protect the ends of DNA from like nuclease digestion and stuff like that. Okay. But more importantly, it also protects them from this concept of what's called gradual shortening. Because of the lagging strand and that requirement for a primer behind you, when you get to the end, there's no more behind you, right? You're kind of like you reach the end of the cliff. And so what happens then? You basically lose that little bit of DNA. We'll see an image for this. It kind of explains it really nicely about how you get the shortening with the lagging strand. But the problem is, after every round of replication of the lagging strand, it shortens a little bit, which pretty much basically means that if it shortened with every round of mitosis, then what would your DNA look like now? You probably wouldn't have any left, right? Because basically you would have shortened your DNA over these millions and billions of rounds of mitosis that you've gone through since the time you were a single cell to now, right? And you basically would have just chewed your DNA up to nothing, right? It would have shortened up to nothing. So that's the problem. So this is what it looks like. So here's our lagging strand here. So notice you have this one last primer, right? That's toward the end that you're able to launch off of. But guess what? That primer is RNA, right? So what happens? Pol 1 comes in, removes a primer, but guess what? Pol 1, being a polymerase, needs a 3' OH, right? Doesn't have a 3' OH? No, because there's nothing left out there. So what you have then is this big old overhang, which we all know the fate of single-stranded nucleic acids, gets chewed back. And what you have then is a shortened template. So if that happens with every round of mitosis, then eventually what's going to happen is you're going to start chewing into some pretty essential genes. And you're going to be running into some massive genetic problems. Okay. So what's our answer to this one? Telomeres. Right. So a telomere basically is a long series of repeats, like an adapter. And it's created by an enzyme called telomerase. And telomerase doesn't have the same problem that polymerase has, because remember, it's an RNA polymerase. It actually has its own template. So telomerase is a self-starter. Because it's got its own internal RNA template, it means it can just basically use that to lay down whatever it wants, right? So oftentimes then it'll use this template and it'll just add. It's just a series of sequence repeats is what it is. And it just basically adds these repeats. And so the telomerase actually has the effect of lengthening the telomeres. And basically what it is it gives you something to shorten. So you just kind of keep shortening your telomere, but your telomerase extends it out so that you never actually get to start chewing up your own DNA. Okay. So it's kind of like designed to be that way. Now, typically speaking, telomerase has been shown to be directly proportional to senescence, cell senescence, right? So length of your telomere and senescence are directly proportional. The longer your telomere, the more longevity the cell has. Now, it's not a one-to-one relationship. There's a lot more going into that whole story of senescence than just telomere length, but there is clearly a telomere length component to it. Why? Because when we've artificially lengthened telomeres, we've extended the shelf life of cells, their ability to basically divide. So telomeres basically actually can be used as a regulator to set your shelf life. So if a cell only has a certain number of mitotic divisions in it, you can set that number with your telomere length, right? When you run out of telomere, then you're out, right? So it's a good way to kind of control your cell population and to sort of manage the population of cells. The idea being that obviously if you've got short telomeres, you're only going to divide a couple of times and you're going to die. Okay. Well, here's the problem with that. Cancer cells don't like that, do they? Because remember, cancer cells fundamentally don't like to be told what to do. We learned in chapter 10 that one of the main genes that cancer cells like to take out is the adult in the room, right? P53. That's a common one. There's another one actually that they like to play with. In this case, they're not taking it out, they're actually turning it on. And that's telomerase. So because of this relationship with cell senescence and your ability to sort of hardwire shelf life of a cell into it with telomerase is very, very carefully guarded and regulated, right? It's like, it's like the tree of life, right? So it's like, you better, before you eat of that one and get immortality, you better make sure that your life is a good one, because the worst thing to happen is to have immortality when your life sucks, right? Because that basically means your life is going to suck forever, right? So something like that would be heavily guarded, right? You don't want to mess with that one. So telomerase is very, very tightly controlled. However, cancer cells love to go after this one and they love to turn it on. So they will actually activate telomerase. What does that do? That will lengthen your telomeres, which will do what? Extend the shelf life of the cells and allow you to basically divide as much as you want, right? That basically is something in cancer progression that we call immortality. Most cells can divide a few times and then they'll die, thanks to the telomeres. But when cancer cells become immortal, they activated telomerase, which means they can divide as many times as they want. And that's a bad thing, right? Because those cancer cells that are accumulating mutations will continue to accumulate mutations, and there's nobody around to tell them to stop, right? Not only is P53 not around to tell them not to do that, but now they have immortality. They can basically divide without bound forever, potentially. Okay. So that's a very important one. So this is kind of how telomerase works. So you can see you got your overhang here. That's from your leading strand. And so what it does basically is it'll grab onto this overhang. And typically that overhang is something that is already in place. That's like a piece of the telomere that's left over, that's shortened. And so you can see this internal template. That's this sort of gold piece of it here. So basically you kind of bind onto that little overhang before it gets chewed up. And then you just use your own internal template to add all the rest of this. And so you're just extending that out. So now instead of this one repeat, now you've got all of these. So you're adding up all of that. And so you can just slide down, grab onto the next terminal piece and just keep on adding. So you can basically just add as many of these little adapters as you want. And what that'll do is that'll just make sure that you have plenty of telomere length to chew up when you go through mitosis. Okay. So an important function, you're basically keep adding or building that adapter set on the tip of your DNA. Okay, so next logical progression, now that we know a thing or two about replication, is what happens if we screw it all up? Well, which we do, right? So obviously we have a couple of things here. That's the DNA repair piece. The first one is obviously our proofreading. So that's our backspace piece, right? So basically if replication flubs up a little bit, you do a mismatch or something of that nature, you catch it. What do? You hit the backspace button. That's that 3' to 5' exonuclease activity that all those polymerases have. So they can backspace up, delete all that and sort of lay it down again. Chances of you making that same mistake twice is rare. Okay. And so that proofreading catches a lot. However, what happens if you have damage that's not caught by that proofreading? Well, typically speaking, generally that usually is not caused by just errors in replication. Most errors in replication will be caught by the proofreading piece. It's like your polymerase will know that it mistyped the word "the," which is one that I can never get right the first time for some reason. It's just like the bane of my existence. right? So that proofreading will be like, okay, I did it again. I typoed up the, so I'm going to backspace it and I'm going to lay it back down, right? So that covers most of the replication errors. What we're talking about now are insults from outside, right? That's basically referred to as mutagens. So this is essentially any kind of a thing that will increase your mutation rate above background level. Now you have a natural background level radiation. We just have things that we're exposed to in the environment, cosmic radiation. We have UV radiation. We have chemicals that just are a natural part of our diet, things of that nature, or in our environment. That's just natural background levels of radiation. So when we're talking about mutagens, we're talking about something above background, right? So typically that comes in two sources, either radiation or chemicals, right? So you have your natural background radiation. That's the reason why we're accumulating mutations over a lifetime. But that accumulation rate is supposed to be very low because background, for the most part, never changes, right? What we're talking about here is spikes in that. So we're talking about, you know, getting too close to the cesium chloride source, or, you know, being part of a nuclear event of one stripe or another. Or in Colorado, it's finishing your basement and living in your basement without doing radon mitigation because you've got open crawl space in the basement. Right? So radon is a radioactive element that's emitted as a gas that is probably one of the primary concerns radioactivity actively in Colorado, right? Because it's in our soils everywhere. And a lot of houses have problems with it. And it's an easy one to get rid of. Basically, you just set up a radon mitigator, which is like a ventilation system that just ventilates the soil in your crawl space out to the outside. And it's not a problem outside. It is a problem in your basement, however, because the gas just traps in the basement and accumulates, and it actually is what causes higher frequencies of lung cancer in Colorado than you would normally see, right? So for us, the radon situation would be considered above background. Okay, just general radon emission when we're out hiking and just walking around Colorado because it's ventilated in the atmosphere, that would be like normal background exposure, right? Just remember that whenever you buy a house and it's got a finished basement, find the crawl space and check for radon mitigation. So, because radon mitigation is not cheap. So that's one of those things where insist that the seller install it, which is what we did. So, so what do we do then if we incur some of this damage? Well, there's a couple of different ways we can do this. There's several DNA repair. If you take the genetics class, we have like an entire chapter on repair. It's actually quite a fascinating discussion because there's a lot going on there. But generally speaking, for our purposes, we can break it into specific or non-specific types of repairs. Basically, in a specific type of repair mechanism, you're looking for a very specific type of damage. So you're very targeted in what you're going after. In a non-specific, it's more general. You're just looking for general anomalies. So you're not looking for a specific type of DNA damage, just anything that looks like it's not right. Okay, so you're just looking for anything weird. So let's take a look at a type of specific repair first. One, probably the most common one we see is photo repair. And a photo repair, basically, this is one that's caused by UV radiation. So anywhere in your DNA where you have two thymines together, which happens quite a bit actually, what will happen is the UV radiation will actually come in and hit those thymines. Now those thymines are supposed to be linked to adenines, right? And paired up with them. But what happens is when UV radiation comes in and hits these, it actually forms a covalent link between the thymines, and these thymines kind of create like a little bit of what's called a thymine dimer. So it dimerizes those thymines and basically eliminates your ability to normally complement with the adenine. Well, here's the problem. When you kind of go through this, your replication machinery doesn't know what to do with this, right? Because they can't do anything with the thymine dimer. And they see the adenines, and so they're trying to figure out, well, what do I put here? Like for instance, if this is your template where the thymine dimer is, and you're looking at that and you're thinking, okay, what do I do, right? Oftentimes you'll just drop in a random nucleotide in there, which will create mutation. And so this covalent linkage is something that has to be broken. Now in bacteria, there's an enzyme called photolyase. It's actually activated by light. It's actually blue light activated. And what it'll do is that'll come in and actually cleave that covalent bond and restore order. Looks something like this. So here's your DNA. Your UV light will come in here, create your thymine dimer, so you can see these little covalent linkages, creating that sort of abnormal bubble up in the backbone, right? So your recognition, your enzyme can recognize that anomaly. And then photolyase, which is activated by blue light, will come in there, cut that covalent bond and smooth it all out. No harm, no foul. Nifty trick, right? Guess what? This doesn't happen in humans. So the question is, well, great. Here we are out there sunbathing, absorbing all this UV radiation, dimerizing all of our thymines. We've got photolyase, right? We can just smooth it all out. We've got some sort of an enzyme that cuts it all up and or makes it all right, right? Wrong. We don't have photolyase. We actually don't have a way to deal with this. And actually in the genetics class, we actually talk a little bit about some of the mechanisms of how your systems deal with these thymine dimers. First of all, in humans, this is unresolvable. You will never get rid of them. They will always be there. And when you go through replication, your cell has to do all kinds of weird little genetic tricks, like recombinational tricks and sort of using, sister chromatids in order to figure out what was supposed to be there. And that dimer never goes away. You just sort of figure out how to sort of jerry-rig yourself around it. And sometimes that jerry-rigging works and sometimes it doesn't. And when it doesn't, that's what creates mutation. And if that mutation is in a cell division gene, that's what creates the cancer. Right? So that's the reason why you want to be careful about your UV exposure, because once you get those thymine dimers, you're living with it. Right? So you can't really remove those. Now, what about a non-specific type of repair? So a non-specific repair, where this is just going after general DNA damage. And so basically what will happen is all of these repair systems have kind of a similar sort of a strategy, a three-step strategy. First of all, they have to have the capacity to recognize the damage. They have to be able to identify this as abnormal, right? And that's true for all of it. And then once they identify it as abnormal, then they have to have a domain of them or a member of their complex whose job it is to actually remove or excise the actual damaged region. Not unlike Pol-1 does to primers, right? You just basically remove it. And then once it's removed, then you lay it back down. You relay it. So the idea is whatever damage was there, if you remove it and you lay it back down again, you do it again, you're not likely going to make the same mistake twice. Okay, so that when you lay it down, then that would be normal. So this is what it looks like. So here's some sort of damaged area. You can see it's always a distortion, a physical distortion of the backbone usually. And then what will happen is your excision repair complex will come in, recognize that damage. In this case, it's a common one that we saw in bacteria called UVR, or ultraviolet radiation. That's what UVR stands for. There's three members of this complex, proteins A, B, and C. So what it'll do is it'll actually bind to the source of damage. And then what's going to do is going to cut around that damage and basically remove it. So that leaves you a gap. Then what happens is a polymerase will come in and then seal that gap, and of course ligase will seal the nick. Okay. And then once you do that, then the idea is like, okay, it looks good now. Right. So this is kind of more like if you were, instead of like just backspacing on a typo, this would be something more along the lines of like maybe rewriting a sentence that sounds maybe syntactically off or something like that. That'd be kind of what this is. Chances are you're not going to be syntactically off twice, right? If you catch it the first time. Okay. So some other types of, there's multiple other types of pathways, all of which are fascinating, by the way, but you can break them into two major groupings, the error-free versions, right? So for instance, if you have a DNA breakage, so if you just have a double strand break, for instance, in your backbone, then you could just seal the nick, right? And there's no error there. That's usually a pretty easy one. Versus error prone, this is basically when you have lots of damage. So likely, in this case, not going to repair all of it. Which basically means that you're probably going to have errors in there, right? So the more you have to do, the more opportunities you have for errors. That's the reason why they call it error prone, right? So, and so that's just the case when you have lots and lots of damage. Okay, so you'll be happy to know that that is the end of 4. At 14, rather, not 4. 4's been over for a while, by the way, if you're wondering. I'm just saying. Yeah, can we redo 4? Good memories of that one. Off to 15 now, or I guess in this case 5, right, if I follow that convention. Okay, so what's the next major mechanism? Well, now the next major mechanism is going to be looking at genes and how they actually express themselves, right? So when you take a look at genes and how they work, you're talking about gene expression. And so the way a gene expresses itself is through making its protein. That's basically what we look at. So the question is, how do we make that protein? And so that's the idea of this one. So largely this could be thought of as the transcription translation chapter. That's basically it, okay? However, as we do with all chapters, we're going to start from the beginning, right? So we always start with our little history lesson, right? Never miss an opportunity to learn a little bit of history, considering how woeful most Americans are at history, like comically woeful. Like I don't even know who the president is now, woeful. But anyway, I'm just saying. So we go back right to the beginning, because remember what we said is that the early part of the 20th century, we were rediscovering Mendel at 1902. This is when William Punnett and William Bateson were rediscovering Mendel. This is when we were working with 4 o'clock plants to see if we did Mendel there. Some people were working with corn, some people with fruit flies, some people were working with humans, right? So we saw a little bit of that before as well, but Archibald Girard was working with humans. And remember in 1902, we didn't really know a whole lot at this particular point. So we were just rediscovering Mendel, still didn't really know a lot about what was going on with chromosomes. The idea of mitosis and meiosis and cell cycle was like really, really cutting edge stuff at this particular point. So we really weren't really that far beyond Mendel at this point still. Because a lot of that stuff didn't really start locking into place until maybe about 6 to 10 years before this with Walter Fleming. But Girard basically was aware of Mendel, and he was also studying a particular disorder in humans called alkaptonuria. Now, the one thing that he knew about alkaptonuria, which is a metabolic disorder, is that he knew that individuals who had alkaptonuria lacked a particular enzyme. Now, I doubt that he actually understood enzyme the way we understand enzyme as a biological catalyst, right, as a protein. He probably understood it to be perhaps a protein that was involved in this particular trait, right? So the nice thing about metabolic disorders is that reactions are very discrete. You've got products, you've got reactants, you've got products, and you've got an enzyme that drives it, right? So at its simplest, it's a very simple structure. So what Girard noticed was that the absence of this enzyme that drives this reaction is what's underlying alkaptonuria. So he kind of had a sense of that enzymatic concept. But what he also noticed is because he was well-read and he read Mendel's work and he was reading what William Bateson was finding and what William Punnett were finding when people were working on corn and 4 o'clock plants and things like that, and they were looking at Mendel's stuff. So what he noticed was that it was actually showing a recessive pattern of inheritance. How do you know that? Because he looked at Mendel's work He saw the monohybrid work. He saw what Bateson and Punnett were doing and others who were doing the similar sorts of crosses in their systems. And they were starting to pull together this concept of this is what a recessive pattern looks like. And he noticed that same pattern in his Alcaptonaria families. Now, did he do what Mendel did and create true breeding strains and cross them together? No, because most people don't like that. Right? We're very picky about who we mate with. I mean, right? That's the reason why we're not in Hardy-Weinberg equilibrium, because we don't do the whole random mating thing very well. Right? So basically what he had to do, he had to basically sit down and get to know families, didn't he? Who in your family has Alcaptonaria? The kids, the siblings, what about them? The cousins, what about them? The parents, what about them? Right? The uncles and the aunts, what about them? Grandmas and grandpas. How far back can we go? Right? So basically what Gerard had to do is basically build a pedigree, a family tree. But in this case, instead of saying, oh, well, you know, I'm related to this person, it's not a genealogical thing. He's mapping this genetic disorder, this human genetic disorder onto that tree. That's how he caught the recessive pattern. Because he saw in his family a similar pattern to what he was seeing in Mendel. And that's how he was able to make the connection of recessive patterning. Really, in a lot of ways, this is sort of like the beginning of human genetics, which we do very differently than stock regular genetics, right? Because we can't manipulate the human system. So we kind of take what we have, and then we try to use other systems and the patterns in those systems to understand something about these families. Gerard is doing exactly what we do today in human genetics. We've learned a lot about patterns of inheritance in other systems, in peas, in corn, in fruit flies. And what we know those ratios, we know those frequencies, we know how to predict them and how to calculate them in those populations. And then what we do is we create a family tree. It's like, okay, I can't manipulate your crosses, but I can take the one that you've generated yourself, right? I can't take your entire family history, and then I can look for those patterns that I now know about, those patterns of inheritance, and I can look for those in your family. And that's the way we do human genetics. That's exactly what Gerard was doing. He was the first one, by the way, to actually make this connection between what Mendel saw and enzymes. That was an important connection, right? Because this basically hypothesizes or seems to insinuate the role for genes, what genes are actually doing, right? Nobody knew that before. Remember, at the same time, we were still asking that question, what is a gene? Don't know. At this point, we don't even know what the genetic material is. This is pre-Hershey Chase, right? So we don't even know what that one is yet. So we're still arguing about this. But somebody like Gerard in human genetics, they basically are doing a lot of this. So now let's go ahead and fast forward a couple of decades, several actually, to 1941. Of course, our history lesson. What's famous about 1941 in the US? December 7th, 1941, a day that will go down in infamy. That's actually a direct quote from FDR. That's Pearl Harbor, right? So basically we were just sort of like putzing around before then. And then of course, Pearl Harbor essentially threw us immediately, instantaneously overnight into World War II, which from most of my studies, Europe was quite happy to see because they were largely getting their tails kicked. I think at this particular point, I think Hitler was learning French and the French were learning German. So, and I think he was getting ready to bomb the daylights out of London. That was the next step. And so things were looking grim in Europe. So there's that. A lot of historians like to play the what if thing. I love those what if games that historians play. What if the Japanese hadn't bombed Pearl Harbor? Would we have stayed out of the war? And what would happen if we did? A lot of historians think that if the US had maintained its distance in the war and hadn't entered at this particular point, that probably Hitler would have been successful. And that his next target was us. right? I mean, I think his master plan was bring the Soviet Union to its knees, and then we were next. So it would have come up to our shores eventually. So here we come. So Beadle and Tatum were important, and what they did basically is they kind of took a slightly different twist on their studies. So up to this point, what people have been doing is studying their natural traits, right? Like for instance, Mendel, all he did was just take his pea plants, and he said like, well, I've got purple and white, so I'm just going to play with those. I'm just going to do like a little bit of horticulture, and I'm just going to sort of tease out these variants, right? So he didn't really do anything. He was just manipulating what was already there. But what we knew was that, especially at this particular point, was that alkaptonuria and other types of disorders of that nature represented sort of a foul up, if you will, of the normal gene, right? And so the big question was, are these foul ups obeying the same genetic rules as the regular gene, right? So for instance, if we were to take the purple pea plant allele and mutate it to fluorescent pink, would that still segregate with the same pattern and the same rules that Mendel saw, right? And that's basically what Beadle and Tatum saw. And what we actually saw was the answer to that was yes, which is quite helpful because that opened up a whole bunch more possibilities. So what they did was they created mutations, and they saw that these behave similarly. To what we refer to as wild type. So for Mendel, purple would be wild type. The mutation which causes fluorescent pink would be the mutant, the pink mutant, right? That's not a thing, by the way, so don't go looking for fluorescent pink pea plants. That just doesn't happen. Okay. But why is this important? Well, it's important because now all of a sudden we have two different things. We now have the capacity to blow stuff up genetically, which is really good because the way we learn the best is by blowing something up and seeing what's wrong, right? That's how we learn stuff. Like, what do you do in this whole process? Well, I don't know. Let's eliminate you and see what happens. We'll take a look at the absence of your function, and we'll learn something about what you do by your absence or by how everything else gets fouled up, right? That's how we do things. So mutation actually was the first time we could create that. We can get in there and just screw stuff up and then see how it changes. Okay. The other thing is that we can also accelerate our studies of these patterns of inheritance because now instead of waiting patiently for variation to occur, we can actually impose our own variation, if you will, by creating mutation. And we can follow that. So we can create our own phenotypes. Instead of waiting for the phenotypes that are given to us, we can now for the first time create our own phenotypes and follow them. They're mutant phenotypes, but we can create those and follow them. And that's exactly the link that Beadle and Tatum were trying to connect, essentially, is this connection between the physical reality that's associated with the genes and these mutations and their behavior being the same in terms of their patterns of behavior. So what they were working with was Neurospora crassa, which is a big fancy word for bread mold. That super simple, super easy, super helpful, a very interesting thing. So what they did is they used X-rays to damage the DNA. So they did what's called a mutagenesis. We do this all the time. So we'll actually take an organism and we'll intentionally blast it with some sort of a mutagen, radiation or chemicals, to create high mutation rates. So it's random, usually it's just mutating everything. And then what we have to do is we have to screen all those mutations because we don't want all of them, right? Some of them are mutations and stuff that we don't care about. And so we have to kind of screen for mutations in the process that we're interested in, okay? So there's a background effect. So this basically will mutate all genes randomly. So whenever you do a mutagenesis, you have to look for something specific. You have to have a selection process. And you're looking for changes in your phenotype. Here's a good example. When I was at UC Davis, I was working on a mutagenesis screen. we were looking for mutants in meiosis. So one of the big key phenotypic markers for meiosis problems is non-disjunction, right? When you get that unbalanced segregation. So what we did was we mutated the fruit flies, and then we screened and mated all the females together to wild type males. And what we did, we screened them for signs of non-disjunction. So we looked for aneuploid flies, polysomic flies, monosomic flies. That suggested that there was a meiosis mutant in that particular thing, especially if it was non-disjunction well above background. And then all of a sudden the question was, what's that mutant doing? Why is it a meiosis mutant? And then we start studying that mutation and figuring out something about the process of meiosis. So that's exactly how you do a mutation screen. Now, what happened here? So what they were looking for was nutritional mutants. Now, one thing to understand about Neurospora is Neurospora, basically, these are a type of organism that can make their own amino acids. Unlike us, where we have to consume our amino acids, they can make their own amino acids. Now, that's normal, but when we mutagenize them, and if we mutate a gene in one of those amino acid production genes, then it will no longer have the ability to make that amino acid. And so we can select for that. So normally what we do is we'll plate these guys on what's called minimal media. This will support growth of healthy individuals. That means you can make your own amino acids, all of them. Now, if you have a mutant in an amino acid, then you're likely not going to live here. Why? Because that amino acid is critical and we don't supply it to you. Right? So we don't give it to you. So if you are a mutant in an amino acid that's critical for survival, then on minimal media, you will die. If you're normal, you'll live. So what we can do is we can use this setup to screen for who has mutations in these critical amino acids. Now, what Beetle and Tatum were looking for is they're looking for enzymes actually that were in the biochemical pathway for arginine production. So they were trying to mutate any gene involved in the arginine production pathway. Okay. And so what they did was they identified some of these mutants, and then they kind of took a look at them and their relationships to each other. So what Beetle and Tatum did was they modified what Gerard implied in terms of the gene enzyme connection. And they basically said that a gene will essentially be driving one enzyme. So one gene, one enzyme. Okay, that's kind of where we started there. That eventually got modified over the 20th century, but that's kind of where it all started. And that kind of is in harmony with Gerard. So this is what it looks like, what Beetle and Tatum did. So you take your wild type Neurospora, you blast it with X-rays. Remember, you're randomly mutating all of these genes. And then what you do is you grow it on rich medium. This is like nutrient rich. It supports everything, right? So this is a situation where I give you all those amino acids. So if you're deficient in those amino acids, because I just blew you up, I blew that gene up, doesn't matter, because I'm giving it to you in the rich media. Why are you doing this? Well, this one is for recovery. Every time you do a mutagenesis, you always follow it with a recovery. Why? Because mutagenesis is harsh and there's a high lethality to it, right? So the idea is you want to blast it with enough X-rays to mutagenize those genes, but you don't want to have so much X-ray that you kill the organism. So what you do is when you mutagenize them, I mean, you just beat the devil out of these things. I mean, they are just like on their last leg. They're like, whoa, I'm about ready to go down. And so what you do is you put them on super rich medium, give them everything they want. The entire Thanksgiving spread. I mean, you name it, it's there, right? So you want it, I'm going to provide it in spades, right? So you have everything you should possibly want. Absolute lack of luxury. So the idea is, you know what, you're beaten up pretty good. So we're going to allow you to sort of heal. Guess what, though? You're still holding on to those mutations that I just introduced in. But as an organism, you're rebuilding your strength. You're beat up pretty good, right? But you're rebuilding your strength. We do kind of a similar thing with chemo patients, right? We basically take a very, very aggressive cytotoxic witch's cocktail, right? Blast it at the patient's body. And if you've ever seen anybody after chemo, I mean, they are wiped out. I mean, they are like on a thread. But then what we do is we basically allow some recovery time after the fact to allow that person's body to sort of regain strength again before you hit them again, right? And that's, you have to do that with multiple rounds of chemo, but this one you don't hit them again, right? It is what it is. You got what you want. You just want them to rebuild, right? So it's very similar situation. So now you've got healthy cells, hopefully, that have been rejuvenated on this rich lap of luxury media. And then what you do is you switch them. This is your screen. So once you've recovered them, now you're going to screen them for your desirable phenotype. So what does that screen look like? Well, you're going to put them on two different types of media. The first one is on minimal media. Now, remember, we said that if you are a mutant of an essential amino acid gene, then you're not going to make that amino acid because it's mutated. So on minimal media, guess what? You die. So you don't get any growth on minimal media. They're looking for that, right? That tells you it's an essential gene. It doesn't, however, tell you that it's arginine, right? Because this could be a mutation in lysine. It could be a mutation in the phenylalanine pathway, right? It could be any essential gene would give you no growth on minimal media. So it doesn't tell you anything about arginine just yet. So how do you know that this is an arginine mutation, not just some other essential mutation? Well, what you do then is you do a supplement, a screening supplement media. So you take minimal media and you add back the arginine. This is what's referred to as a rescue. The idea being here is if arginine is what you're missing that's allowing you to die because you don't have it, then if I supply what you need, you should live, right? So if arginine is not the problem, guess what? You'll still die. So they took a look at this and they said anybody who grows on arginine supplements basically means that arginine was the thing that you were missing, right? So we were able to rescue you. So these are all arginine mutants. By the way, notice these are all arginine mutants. These would all be true for enzymes, regardless of how many there are, right? We don't know how many there are at this point, but any enzyme involved in the production of arginine is going to show the pattern, okay? So notice you didn't say, oh, I've got one gene or two genes. You haven't done that one yet. You're just saying whatever this is, it's actually involved in the arginine pathway and it's necessary. That's all you've said, right? Now, how do you know how many are involved? Well, that's where we come down to this one. So what they've done is, first of all, biochemically, they knew that the pathway to arginine contained several intermediates. For instance, you started off with an amino acid glutamate, which they knew was turned into ornithine, which was then turned into citrulline, which is then turned into arginosuccinate, which then finally was turned into arginine. Wow. Okay. So if that's the biochemical pathway, then the question is, how can you figure out what's going on in there? So what they did was they essentially did these results. So essentially successive additions of your intermediates, kind of a rescue experiment, kind of like a successive rescue. So what they did was, if you added arginine to your media, guess what? Everybody grew. Good. That means everybody is involved in the arginine pathway, okay? So now if they added arginosuccinate, they knew that arginosuccinate was the precursor to arginine. So that means in this particular case, if you added this, then you had everything else that you needed in order to finish out, right? That means whatever that enzyme is right here, you finished out. So what they noticed is you got growth, good, that's a healthy enzyme. Growth, that's a healthy enzyme. Growth, that's a healthy enzyme. No growth, whoop. That means this guy right here is lacking that terminal enzyme because it's not able to finish out. They did the same thing. So they said, okay, let's add citrulline. Same basic idea, right? If these two enzymes are healthy, these two right here, then you should see arginine and growth, right? So here you see arginine growth, and then all of a sudden, you don't see growth anymore. So what they're saying here is that at this particular point, you've got a mutation in this point because you can't make any arginine. It's an obstruction to that pathway. Same thing with this. If you add ornithine, you can say if all of these are 1, 2, and 3 are healthy, then ornithine should produce arginine. That's only true for the first one. The rest of them, no. Right? So they knew that this one here was early in the pathway. This one here was later in the pathway, and then this one here was the latest in the pathway. Of course, that's only the beginning, right? Because then we still have glutamate to ornithine, which is the first one in the pathway, right? So they were able to do by doing this, they were able to order what the pathway was, like who was first, who was second, who was third. Before, all they had was a bunch of intermediates. They didn't know who came first or who was second or who was third. By this one, they were able to order it. So you can see here that basically your arginine gene, which they mutated, eliminated your H enzyme, which is what gave you the all growth on arginine. Of course, if you're mutating here, this gene, you're mutating and destroying this protein, which blocks you here. So you can add all the citrulline you want, but you're not going anywhere. You add arginosuccinate, and then you're going to finish out. Okay. Same thing here. If you mutate this enzyme by mutating the gene, then you're blocking this piece here. So all the ornithine you can add is going nowhere. But if you add citrulline, you'll finish out. If you add arginosuccinate, you'll finish out. Okay. And then of course with E, right, add all the glutamate you want, you're not going anywhere. But if you add ornithine, then you'll finish out. If you add citrulline, then you'll finish out. If you add arginosuccinate, then you'll finish out. So basically what Beadle and Tatum did was really pin down this connection between genes and enzymes. One gene, one enzyme, because what did they mutate? They mutated the gene. And then they saw a disabling of the enzyme itself. They were also able to illustrate how you determine whether or not you're dealing with a biochemical pathway. So they showed us how to study biochemical pathways. Which before this, we thought that, one reaction, one protein, or one process, one protein. We didn't think that a particular process could be like a multiple step system, that it could be a pathway of multiple steps that all relied on each other. Beadle and Tatum did that for the first time, and they showed us how to tease that out, how to determine whether or not you've got a single enzyme or if you've got multiple enzymes that are in a biochemical pathway in a stepwise process. That became critical because, as we now know, the majority of what you're doing in your body is some sort of a pathway. There's very few things that are just one step, boom, and you're done. Most of it is multiple steps in a pathway. Okay, so that brought us then to the 50s, where in the 60s, where we have this concept of central dogma, and this is kind of where Crick, of course, we all know how we feel about Crick. But to Crick's, he did do some functional things. So it wasn't like he was a complete schlub. He wasn't, out there surfing and smoking marijuana on the California coast. Right. So he actually did make some meaningful contributions, especially in this particular field as well. He's the one who came up with the idea of central dogma. And it simply is describing the flow of information, that the flow of information in a cell moves from DNA to RNA to protein in that order. And your conversion from DNA to RNA is transcription, which I abbreviate XPN. So if you see that, that's transcription. And then from RNA to protein is translation, which I abbreviate as XLN or TLN. Now, there are some violations to this, right? So some viruses which have an RNA genome will go through what's called reverse transcriptase. So it's a reverse transcription. So you basically make a DNA strand of an RNA copy, and then you make a DNA. So you go from RNA to DNA. So that's a little bit of a violation. But for the most part, this is your central dogma. This was before we really understood retroviruses, actually, when Crick came up with this particular concept. So here's kind of a picture version for those of you who like pictures instead of words, right? So basically, you got your DNA template. You go through transcription in order to get your RNA. And then from your RNA, you use that code in order to inform you as to which amino acids to select when you're making your protein in the ribosome. And so that's what this chapter is about. This chapter is all about transcription and translation. That's the reason why we start there. So let's take a look at it. Transcription. So basically, when you take a look at transcription, you're going to be making an RNA copy. And so generally speaking, you only have one strand of DNA that's going to be copied at a time, not both of them. That's replication, right? And then, of course, the other thing is when you're making RNA, you're going to be trading out your thymines for uracils, right? So wherever you see an A in your template, you're going to be adding a U, right? So that's the common thing. By the way, there's a couple of major things you can tell you're dealing with RNA. Number one, it's single-stranded, right? Number two is if you see a bunch of U's in there, you know you're working with RNA. Okay. And then this is what then gives you the information that you need for making proteins. And we're going to see how that works here when we get to translation. Okay. Now, translation specifically is focused on production of your polypeptides, right? So it's basically selection of your amino acids. And then you have three different kinds of RNA are required to do translation. Okay. And we'll take a look at the role of all three. But this is just like the basic intro, transcription and translation. So we're going to get much deeper into the mechanisms and the details of transcription and translation here in just a second. So let's take a look at RNA. So since we're talking about RNA, right, and that's what transcription does, is you're making RNA. What exactly is it, and what does it do? Well, basically, all RNA is a copy of DNA. That's basically what RNA is. You don't make it on your own. It's a copy of DNA, which basically means you always have to have DNA template in order to inform your RNA. But what does it actually do? That's great. That tells me what it is, but it doesn't tell me what it does, right? So one of the things that we learned in the 50s, 60s, and 70s as we were studying transcription and translation is that generally there's three really dominant types of RNA. It used to be back in the day that these were the only three that you should mention, but we've learned a lot about RNA since then. Is messenger RNA is one we have. So this basically is the code for the message for selecting your amino acids during translation. So you want to know which amino acid to select? Well, guess what? That information is going to be found on the mRNA. rRNA is, and we have talked about this off and on throughout the semester, but this is a structural component of the ribosome. And so it's involved in translation by being a piece of the structural component of the ribosome itself, during ribosomal assembly. And then tRNA is essentially the translator that converts the mRNA code to amino acid. Right? This is the Rosetta Stone. Anybody know what the Rosetta Stone is? Oh, here I go again, getting outside of the lines of the discipline. You've probably heard of it, right? Most people will say, like, isn't that like a program that you buy to learn Spanish or Chinese or whatever? You've heard of that one, right? Buy Rosetta Stone, learn Spanish in 2 days. No, not that one. Right? The actual real Rosetta Stone. It's actually a significant moment in archaeology. Oh, here he goes again. We're off into archaeology, right? Grab your pickaxes and your brushes because we're going digging, right? No, what's the Rosetta Stone? The Rosetta Stone is important, right? Because what the Rosetta Stone was back in the 19th century is in Egyptology. we were fascinated by Egypt because we didn't know a lot about Egypt. And we had all these like temples and like Karnak, for instance, and all these things with like and tombs. We're discovering tombs with like all these pictures all over the walls. And we realized that these pictures weren't just art, they were actually language. And so we realized that the entire Egyptian language, hieroglyphics, was based on images. And none of the current languages in the world have an image-based interpretation to them. All of ours are symbol-based, but they're not image-based, right? Like, for instance, we'd say like, okay, what does the picture of the cow mean, right? Or like, what does the eyeball mean, right? You know, so we knew they meant something, but we didn't know what they were. So we had no clue what hieroglyphics meant until we discovered a stone in archaeology that had an inscription on it. So I don't even know what the inscription was, it's not really important. But it was actually created in probably either early AD or late BC. It certainly was the overlap between Hellenism, Roman, and Egypt. So it was pre-fall of Egypt, so pre-Cleopatra, probably in the Ptolemy era, in that era. So that kind of post-Hellenism era where you had equal parties. And so this inscription was actually inscribed in 3 different languages. One of them was in Latin because of the Romans. Another one was, I believe, Greek because of the Hellenists, right? So that would put it in the era of the Ptolemies, and Greek was the kind of the language of the world back in those days because of Alexander the Great and the Hellenization of the area. Greek was like the working language, but it was something that it was associated with Egypt. So there was also Egypt's traditional language, which was hieroglyphics. And so their written language was hieroglyphics. And so you had an inscription that was basically triplicated in Latin, in Greek, and in hieroglyphics. And from that, they were able to use the languages that they knew, Latin and Greek, to decode what the hieroglyphics meant. So they were able to assign word values to the eyeball or to the cow or whatever. And so that allowed them to basically decode hieroglyphics. And that was the major event. So this was a stone that allowed you to be able to convert from one language into another, right? Just like a translator. And that's exactly what tRNA does. The molecule is rigged so that it can translate between nucleic acid language into amino acid language, from nucleic to protein, right? So in that sense, it is very much a Rosetta Stone, a molecular Rosetta Stone. So, but in the last 20 or so years, we've discovered a ton of other types of RNA. There's what's called small nuclear RNA, which is involved in the process of splicing, which we'll talk about later. You have small recognition particles, SRP RNAs, which are basically for localization of proteins. You have microRNA, which oftentimes are regulatory. And so the amount of RNA that we're finding is actually mind-boggling. So all these little, we used to think all these little itty bitty chunks and pieces of RNA are largely just like degraded debris from the other RNAs. It's like degraded tRNA or degraded mRNA or something like that. What we're finding out now is that a lot of these little itty bitty pieces of RNA that we kind of overlooked before or just assigned to degradation before are actually regulatory. So they actually have a regulatory function or they have some other type of a function like that. And we're just now starting to discover the broad and deep and wide world of RNA and the function of RNA. It's actually a fascinating area. So then that kind of leads us to the big chase, right? This was another hot, heated chase. It wasn't quite as heated as the structure of DNA, but it was a pretty big one, right? The idea was this, great. So now we know that the mRNA has the code for selecting an amino acid. And we know that tRNA is the Rosetta Stone that sort of decodes that, right? But here's the question. What is that code? Right? What does the DNA sequence mean? AAA. Right? I mean, how is that a code for this? Right? And what is that code? Well, that's what this chase is, right? The chase to the genetic code basically was driven by a lot of people. I mean, certainly Crick was involved in that process early on. Sidney Brenner, my boy, This is the guy who basically developed C. elegans as a model organism, did a lot of work on genetic code. There's a couple of things we had to learn, first of all, in order to be able to define the code. First of all, we had to discover the idea of a codon. What that basically means that the code exists in 3-letter words. Called codons. And that each 3-letter word is essentially assigned to an amino acid. So one codon, that is to say 3 nucleotides or 3 base pairs, equals 1 amino acid. How do we know this? Well, we knew this by working with frame shift mutations. What we did is we basically played around with single and double as well insertions or deletions of nucleotides in a sequence, and we looked for what it did. What it created was what's called a frame shift. And I want to illustrate to you what that means and why it tells us that we're looking at a 3-letter word system. I'm going to just write down some sequence here. So generally speaking, when we are looking at the frame with which you're reading, we're reading this originally as this is being position number 1, 2, and 3. So that's one word. And then we've got 4, 5, 6, that's another word. 7, 8, 9, that's another one. 10, 11, 12, that's another one. 13, 14, and 15. How do we know that? Well, let's go ahead and insert one. Let's insert an A over here. All right, what happens if we insert an A over there? Or if we do 2, what's going to happen? Well, what happens is now all of a sudden this first A is read as 1, this is 2, and this is 3. Now this is 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Now this is 16 over here. Notice what's happened. I've shifted the frame. It's no longer in frame. It's out of frame now, right? Because now instead of AAC, it's now AAA. That's different, right? So it's not reading the same. The same thing is also true about deletion. So what if I happen to delete like that one right there, right? Delete that first one. Well, this is going to be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So now notice what I've done is I've shifted it that direction. I'm out of frame now. Instead of AAC, I'm now ACA is my first amino acid. And so everything makes no sense after that. Right? Now this happens with 1 or 2 deletions or insertions, but not 3. What happens with 3? With 3, all that happens is you just miss the entire amino acid, but everything else is the same. It's like you omit that amino acid, but everything else is the same. So that basically means that there is a 3-letter frame. If you put 1 in there, it's odd, right? So it's going to shift it. 2 in there, it's odd, it's going to shift it. If you put 3 in there, then all that's going to do is change that 1 amino acid, but it's not going to change all the rest of them because you're still in frame. That makes sense? So it's kind of what it looked like. So basically when you see this, you can see here is your original sequence, ACG. If you delete out the C, it's now going to be AGC instead of ACG. So what's happened is now you have methionine and proline, but now you've got basically serine, threonine, alanine. So this is just like, huh? Like that's just all garbage. It's just like randomness now because it's all shifted. It's been shifted from that one deletion. If you delete 3 of them, so if we take out that entire thing, notice what happens is we remove, in this particular case, the threonine is gone, but the histidine and everything else is still the same. So you've omitted, in this case, threonine, but you haven't shifted. This tells us that if we add or delete 1 or 2 out of frame, it'll shift it. If we add or delete 3, it will not shift it, but it'll create either an omission or an addition. But the frame's not shifted. That tells you that you're reading things in 3-letter words in groups of 3. The other big thing we wanted to know is this, does it have punctuation, right? So is it spaced or unspaced? And so what does that mean? So when we have a sentence, we have spaces between the words, okay? And the idea here is, do the spaces count? That's what we're trying to figure out. Or do they not? So if the spaces count, then what that basically means then is if I have like ACC, that's going to be 1, 2, 3. But if I omit that A, it's going to be 1, 2, 3. So it'll count that space or not, right? So if it counts the space, then it'll maintain its assignment as number 1, being that open empty space, right? If the space doesn't count, then what that means is the space doesn't mean anything, and the next major occupied slot is the one that counts. So what did we see? We saw that basically the spaces don't count. All right. So for instance, in the case here, if you have spaces, if you delete the T, then the idea here is you're just going to hold open that space and you're going to read HE, space HE, right? So that's going to be what you read. But what we actually found was that if you omit the T, the space doesn't count. What you do is you just shift it over and you start reading it in your shifted idea. So this was 2 of the questions we had, right? Do the spaces count? And the answer is no, they don't. Okay. Only the bases count. So if you create an opening because of a deletion, then that automatically shifts over to the next available base before you start counting. Okay. So then this obviously then led to a lot of questions, right? Okay, this is great. So now that we know they're 3-letter words, which one of those 3-letter words are for which amino acids, right? So how do you assign amino acids to 3-letter words? Well, Marshall Nuremberg did a lot of this work, and it's very simple experiment that he did. Essentially, he created an in vitro translation assay. That is to say that usually you probably could cell abstract, you know that everything is in there for translation. So you have everything except for your template. That is to say your mRNA. And in this particular case, what he did was he just basically synthesized mRNA with different sequences, different codon sequences in there, put it into the translation reaction, and then he analyzed which proteins he got. So for instance, here's what he'd do. He'd say, okay, let me go ahead and make an mRNA that's like UUU, like all U's like this. I'm going to stick that into translation. And then what am I going to get in terms of proteins? Well, I'm going to get a 3-long chain of phenylalanines. So what does that mean? That basically means that UUU is the assignment for phenylalanine. Now you can imagine then that it's just a matter of just mixing them up, right? Just try different combinations. Try UUA, try UUC, try CAU, all these different combinations, and then stick them in this translation experiment, and you just analyze which of these codons gave me which amino acid. And from that, you can basically assign the amino acid designation to each one of these codons. And so in addition to amino acid designating codons like UUU, for instance, is phenylalanine, he also found that there are stop codons, right? Codons that basically will terminate and end translation. There's 3 of them: UUA, UGA, go Bulldogs, and UAG. So those 3 were stop codons. Wherever they exist, translation stops. So for instance, if you were to do UUU, UUU, UUA, what would that one look like? That would basically give them 2 phenylalanines, right? Because that UUA is a stop. Whenever your stop codon comes in there, it doesn't matter what's before it or after it, it just stops. Okay. He also noticed that there's a start codon, a common start codon, AGU, that basically creates a signal for the start of translation. Every protein, you noticed, has the start codon, which is AUG, which encodes for a methionine. So every protein, actually the first amino acid is methionine. And another thing that he also noticed was, as he was playing around with this, is that the code is what we refer to as degenerate. And that is to basically say that some amino acids have more than one codon. So some amino acids have multiple codons. That code for them. And over time, and there's others who kind of helped out with this as well, he was able to basically assemble what we now know as our genetic code table. And we take a look at our genetic code. Basically, when you're looking at something like this, you can see you're going to analyze, you got your first letter in the downward column. Your second letter is to be read from left to right, and your third column is also down. So for instance, let's take a look at some RNA here. How about this one? So basically, if we wanted to translate this, now this is going to be mRNA here, right? So a UUU is going to be, here's my U, right? If I go to next U and then I have UUU, that's going to be a phenylalanine. What's CCC? Well, here's my C, and then I have to go to this column right here. So it's going to be this guy right here, and the CCC is right there, so it's going to be a proline. AAA is going to be down here. If I move over to the A column, that's going to be right here, and then my AAA is right there. It's going to be a lysine. And then AAC, and back to A again. But now I want to go to A, and then my C is going to be right there. So that's going to be an asparagine. So that's basically selecting out your amino acids. So that's basically how you use the code. By the way, a very, very important thing to do in a situation like this is to basically, now that you know replication, is to test yourself, right? Take, for instance, a sequence of DNA. And then challenge yourself to replicate it. So what's the next strand, right? So that's replication, right? And then pick a strand, any strand, like let's say that one right there, for instance, right? And then do transcription of this strand right here. So what's the complement of 5'? 3', right? In this case, it's going to be U, U, A, C, U, whoops, G, sorry, G, U, C. And this one right here is going to be A, U, and then A, and that's going to be 5', right? That's transcription. The important thing to understand is this is all written 5' to 3', all this stuff. So in order to translate this is what I want you to do, right? Translate this one. You have to flip it around. That's always one thing to keep track of. So this would, you would want to read this one as 5' AAA, UAA, CUG, and then AUU. That's the way you would want to read it. Okay. That's always an important thing to remember, is because by convention, anytime you see sequence, it's always written in the 5' to 3' direction, left to right. And that's usually how you interpret everything. So whenever you get something, and that's the reason why I picked that upper strand, by the way, right? So whenever you get something like that, you notice it was flipped around, right? So you have to rewrite it, flip it around so that your 5' is on the left and your 3' is on the right, and then you can process it. Then you can go in here and say like, okay, what's AAA? Well, that's a lysine, right? What's UAA? Well, in this case, it's a stop codon, right? So we're dead. We're done there. So we got a lysine and that's it. So does that make sense? So what I would do is just make up some DNA, challenge yourself, replicate it, that is make the other strand, and then transcribe it and then translate it using this table as a code. Notice also, I want to come back to this. I don't want to hit this just now because, you know, I don't want us to be like forever late on the lab exam. But there's something I want to come back to called coding strand and template strand. And I've got it written out here perfectly to show that to you, but we'll talk about that one a little bit later. When we start off next time, we'll talk about our cute little piggies. Okay.